Hello, I'm Dr. Iftikhar Kalu, a consultant and professor of medicine in the Department of Cardiovascular Disease at Mayo Clinic, Rochester, Minnesota.
And I am Dr. Safarova, an assistant professor of medicine and a postdoctoral fellow in the Cardiovascular Biomarkers Research Laboratory, also here at Mayo Clinic in Rochester, Minnesota. We are delighted to summarize for you the main findings of our manuscript entitled Variability in Assigning Pathogenicity to Incidental Findings, Insights from LDLR Sequence linked to the electronic health record in 1,013 individuals, on behalf of our co-authors.
Pathogenic variants in one of three genes, that is LDLR, ApoB, and PCSK9, account for the majority of familial hypercholesterolemia cases. The American College of Medical Genetics recommends returning pathogenic variants in these three genes to individuals who have undergone whole exome or targeted sequencing. However, false assignments of pathogenicity to genetic variants detected by genome sequencing can have adverse consequences in clinical practice. It is therefore important to explore processes and criteria for assigning pathogenicity to specific variants that are detected as a result of genome sequencing.
However, information regarding pathogenicity is not known for the majority of genetic variants. In public databases such as the Human Gene Mutation Database and ClinVar, almost half of the variants are left unclassified. 17% of genetic variants interpreted by more than one submitter in ClinVar have a discordant interpretation. When the recommended standards and guidelines are applied, there is still a discrepancy in interpreting variant pathogenicity among genetic laboratories.
These findings reinforce the need for a standardized disease gene-specific quantitative approach to variant classification. And in this paper, we focused on a specific gene, that is LDLR, and a specific disease, that is familial hypercholesterolemia. We assessed discordance in reported classification among submitters to curated databases and among expert reviewers in assigning variant pathogenicity in individuals who underwent sequencing of LDLR in a CLIA-certified facility.
1,013 Mayo Biobank participants at risk to start on a statin therapy within 3 years who were impaneled in the Mayo Clinic primary care practices from 2012 to 2013 and underwent genetic testing using a next-generation sequencing PGRN-seq platform were included in this report. Variant selection in LDLR was completed using bioinformatics prediction tools, variant database review, and expert review using 2 different protocols. Out of 178 variants passing quality control filters, 25 putatively functional pathogenic variants in LDLR were identified in 124 individuals. Manual review of filtered variants was performed by 2 independent specialists, a laboratorian and a cardiologist, both involved in the interpretation of genetic testing results for FH. In the manuscript, we describe in detail the workflow and data analysis for LDLR variant interpretation. One reviewer used a conventional assessment protocol and classification system for variant designation adopted by Mayo Clinic's Department of Laboratory Medicine and Pathology, while the second reviewer annotated variants based on scoring criteria tailored based on the lines of evidence recommended by the ACMG guidelines.
All LDLR variants were rare or low frequency. 60% were missense. As of August 2016, all 25 variants were present in ClinVar. 19 variants had more than one interpretation, and 26% of LDLR variants were reported to have discordant interpretation at the level of clinical actionability. Using a 3-tier classification system, 24% of variants were assessed discordantly by the 2 reviewers. The variability in weighting the impact of available components for variant classification was the main cause of discordance. A final discussion of which of the variants to return led to a unanimous consensus that 2 variants should be returned. In summary, this report describes a framework for a gene-disease tailored approach to classification of variant pathogenicity. We are next going to explore a machine learning quantitative genomics approach for weighting levels of evidence and to assign pathogenicity to genetic variants detected by genome sequencing.
We want to acknowledge the right protocol and Mayo Clinic Biobank study investigators, field staff, and study participants. We hope that you find the manuscript of interest. Thank you for watching.
Thank you.